Okay, I'm going to take you through how to do a plan diagram, one last one. This time I've picked something that's probably totally unfamiliar to you. The image here is actually a cross-section of a root. This is a corn or maize root, but this is not something you're expected to be familiar with. So in this case you won't be asked to label structures, or at least not unless you've covered this in one of the later topics. So let's get into it. So in this case we're just doing a plan diagram, we're not labeling any of the tissues. So just going to show you how to do that. If you're unfamiliar with what the tissues are, how what to look for in terms of what to draw. So first off, we have got quite a circular structure, so I might start with drawing a circle. This makes planning the use of our space a lot easier, because once I've drawn that circle, everything else goes inside. So it's quite a nice one in that respect. So again, not taking my pencil off the page. My circle's a little bit square. They say apparently your ability to draw a circle freehand is a mark of intelligence, so I don't know what this circle says about me, but hopefully nothing too bad. Anyway, so looking at a diagram now, trying to differentiate some tissues out there. So first thing I'd be looking for is with any kind of layers, an outer layer. If we're talking an animal, it might have an epithelial, a skin, or a plant would be epidermis, again basically just the plant skin. And looking at this one, I do think there is an outer layer, a single cell sort of outer layer. And the main reason for that is if you look at the colour, the outermost layer of cells are very white, whereas the ones slightly in from that have stained a lot more red. So that difference in staining is going to be a hint, partly for our tissues here too. So I'll draw the innermost, the outermost, so probably an epidermis. Okay, holding the pen, moving the hand. Doing that once more. Okay, fantastic. So there's my epidermis. Now moving on to my other layers. So around the outside here, what I can see is there's some cells towards the outer edge which have stained very dark red, and moving towards the center, they're much less stained. Now there is arguably sort of a gradual transition there, so I could do a dotted line, or I could just do a slightly fainter line. But I think there's enough of a division there to essentially just draw a line. So let's do that. Okay, fantastic. Alright, and then we've got our innermost circle. We can see a dark red line there dividing that, separating that. That looks like it's sort of the one side of the cells there, or one small layer of cells there possibly. So let's draw that one in. Okay, fantastic. Arguably we should make that a double layer, but it's very thin, and my diagram here is quite small to fit on the camera, so I think I might actually just leave that one. So let's go for the circular structures, these sort of large white circles. So 1, 2, 3, 4, 5, 6, 7, 8. By the way, those are actually xylem. And then we can see there's again a difference in staining between the outer tissue there and the inner one. So just sort of following the edge of these. Okay, and then in the middle again the cells are a little different, so we might just try and indicate that. Okay, and really what I'm looking for is a difference in cell shape, a difference in maybe the thickness of the walls. Here maybe that's a little bit difficult to see, or a difference in staining, and that's probably here. You can see that quite clearly or quite obviously. So I think that would probably be it. You know, labeling structures here, you're really not going to be able to label something that's unfamiliar, and you won't be expected to. So if you're drawing a plan diagram, I think this would be a reasonable plan diagram for a picture like this. Cool.